Today we will discuss about the Vibrio cholerae. What is the general identification of Vibrio cholerae? It is gram-negative comacid bacilli. What is the classification of gram-negative bacilli? Depending upon oxidase enzyme, gram-negative bacilli are divided into two, that is oxidase positive or oxidase negative. Oxidase positive organism, on depending upon O2 requirement, divide into aerobic, anaerobic and facultative aerobic. Vibrio cholerae is under the facultative aerobic. So Vibrio cholerae is under the facultative aerobic, that is it is oxidase positive and facultative aerobic. So now come to the features. First, it is oxidase positive. Second, so dartling type of motility. Dartling type of motility. It is also called shooting star motility. Normally Vibrios are non-halophilic. Normally Vibrio are halophilic. But two species of Vibrios are non-halophilic, that is Vibrio cholerae and Vibrio vulnificus. Those two species are non-halophilic, that it does not love salts. Fourth, high pH is required for their growth, like above 8. On gram staining, fifth on gram staining, it shows fish in steam like motility. Fish in steam or swarm of gnats like appearance. So it is look like fish are swimming in the steam. Okay, so it is called fish in steam or swarm in gnats like appearance. Now come to the culture media. So first is selective media. The most important selective media is TCBS. What is the full form of TCBS? T for thiosulfate, citrate, bile salt, sucrose agar. Normally this TCBS agar is look like green, but fermentation of sucrose causes transformation of the color green to yellow. So what is the indicator of TCBS, so that the color changing is occurring? That is bromothiamol blue. Bromothiamol blue is the indicator. And what is the pH of this TCBS agar media? That is 8.4 to 8.6. So colony morphology in the TCBS media. Here we will find oil drop or water drop like colony. Here is the color of the TCBS is green, but due to the fermentation of sucrose it transformed into yellow color, and here you find that oil drop appearance of colonies, oil drop or water drop colonies. Now second selective media. It is bile salt agar. Here same type of morphology, that is oil drop colonies are formed. Now third, MGTTTA agar. What is the full form of it? M for Monsieur, gelatin, terulite, torocolate, tryptase, agar. So what is the full form of MGTTTA? Monsieur, gelatin, terulite, torocolate, tryptase, agar. So it is also a selective media. Now next, come to the enrichment media. Enrichment media are alkaline peptone water. Only one media, alkaline peptone water. We all know enrichment media is broth, means liquid culture media. So it is alkaline peptone water. Now come to the transport media. Three transport media are present, that is Venkatraman Ramakrishnan media, Venkatraman Ramakrishnan media, Caddy Blair media, Caddy Blair media, and third alkaline peptone water again. Now come to the classification of Vibrio, very important. Name of the classification is known as Venkatraman Gardner classification. Venkatraman Gardner classification. And what is the basis of this classification? Those classification is based on O antigen. And what is the O antigen? O antigen means somatic antigen. Basis of the O antigen, Vibrio cholerae can be divided into three groups. That is O1, O139, and non-O1 and non-O139. These group include O2 to O138 and O140 to O206. Now important concept is, if we are able to differentiate any organism by biochemical property, it is known as biotypes. If we are able to differentiate any organism by its biochemical property, it is known as biotypes. So biochemical properties. So now O1 groups can be divided into several biotypes, that is classical and Eltor. Classical and Eltor. Those are the biotypes, means we will differentiate the organism based on biochemical property. Each classical and each Eltors having three serotypes. Each classical and each Eltors having three serotypes. Serotypes mean differentiating the organism by the antigenic property. So differentiating the organism by antigenic property. And what are the serotypes? That is Ogawa, Inaba, and Hikojima. So O1 groups is divided into two biotypes, that is classical and L-TOR, and each classical and each L-TOR having three serotypes, that is Okawa, Inawa and Hikojima. An important point: in India, 1 to 6 pandemic is due to classical variety, and the 7th pandemic, which is occurred in Indonesia, is due to the L-TOR variety. Now come to the difference between the classical and L-TOR Vibrio. So first is properties classical and L-TOR Vibrio. So what is the first property? That is polymyxin B sensitivity. Classical Vibrio is sensitive to polymyxin B, but L-TOR is not sensitive, that is resistant. Second, O129 antibiotics. What is O129? A bacteriostatic agent obtained from bacteria. Static agent. So it is also sensitive to this O129 antibiotics, but L-TOR is resistant. Third, phase 4 typing. Again it is sensitive, it is not sensitive. Fourth, SIP RBC agglutination. Sorry, SIP RBC hemolysis. It is negative, it is positive. Fifth, CHIK RBC hemagglutination. It is negative, it is positive. That is classical is negative, L-TOR is positive. Sixth, modified CAM paste. Again it is negative, L-TOR is positive. 7, VP reaction. Classical is negative, L-TOR is positive. 8, phase 5 typing. Again it is classical is negative, L-TOR is positive. Remember, Listeria and Vibrio shows modified CAM test positive. Now come to the pathogenesis. Pathogenesis is due to the some virulence factor of Vibrio cholerae. What are the virulence factors? The important virulence factor is cholera toxin. It is also known as CT. It is bacteriophage mediated. It is lysogenic and act on adenyl cyclase enzyme. So what is the consequence of it? Is that it increases the cyclic AMP, which causes the release of water and electrolytes from the intestine, from the GIT or intestine. So it is associated with watery diarrhea, or also known as secretory diarrhea. Next important virulence factor is zona occludens toxin. So this toxin disrupt the tight junctions of the cell. Cells are attached to each other by the tight junctions. So zona occludens toxin disrupt the tight junctions of the cells. So increased permeability occur. So this is also associated with secretory diarrhea. Now third, toxin conjugate pili or TCP. It helps in attachment and pushing of toxin. So help in attachment and pushing of those toxin. So now come to the disease. First important disease is profuse watery diarrhea, also known as rice watery stool. And second is abdominal pain, cramps, dehydration. Why dehydration? Due to the loss of water and electrolyte release. And circulatory collapse and death may occur. And what is the main cause of the circulatory collapse and death? Due to the dehydration and electrolyte imbalance. Okay, now come to the biochemical test. It is positive for COINS, that is coins. What are those? C for catalase positive, and already discussed it is oxidase positive. I for indole positive, nitrate reduction positive, and sucrose is also positive. Sucrose fermentation positive. Now come to the sting test. What is this? If we apply the sodium deoxycholate on the culture of this bacteria, so application of sodium deoxycholate on culture, it causes the denaturation of bacterial protein. So if we take the bacterial colonies with loops, bacteria comes like thread. It is called sting test. So bacteria comes like thread as the protein is already denatured, called sting test. So make a diagram. So let it is the culture media, and here is the bacterial growth. So application of deoxycholate causes the denaturation of protein. So it is let sodium deoxycholate. Sodium deoxycholate, it causes the denaturation of protein. When we take the colonies with the help of with the help of loop, it causes the sting formation. It is the sting formation. So sting test is positive for Vibrio cholerae. Rapid diagnosis can be done by the motility inhibition test. How? Let a sample on a microscope shows the dartling motility. Dartling motility. So it may be Vibrio cholerae. Now we can add antiserum in that stool sample. After 30 minutes, if we observe that under the microscope, then we will find that all bacteria are now non-motile. That is, motility of the bacteria is inhibited. Why? Because the application of antiserum. So it confirms that it is Vibrio cholerae. Last but not the least, ELISA and PCR can also be done for rapid diagnosis of Vibrio cholerae.